Hello everyone, it's 12 o'clock, so we will make a start. I just wanted to start by saying a huge thank you for registering and coming to this event, where we're kicking off the Integrating Health Research Mechanisms to Populations research strand, which is very generously funded by the Elizabeth Blackwell Institute. We're really delighted so many of you have joined us today, and we hope that you'll pass on the word about this strand to postdocs, PhD students, incoming fellows and other colleagues who might benefit. In order to help this run smoothly, I just wanted to start with a couple of housekeeping points. Please can everybody keep their videos off and remain muted. We've got a question and answer session at the end, and during that we'll invite people to unmute. If you have questions for the question and answer session, please do feel free to use the chat function to introduce your questions, and feel free to post them during the presentations, and we'll keep an eye out for those. Please note this meeting's being recorded for the benefit of some of the people that couldn't attend the meeting who requested it. We will make the slides and recording available for attendees following the meeting. What we want to do today is to give you a bit of information about how you can engage with the strands and what we want to be able to do to help you. I'll give you a brief overview of the people you'll hear from today. To begin with, we'll hear from Professor Rachel Guberman-Hill, who's been the director of the Elizabeth Blackwell Institute since 2017. We'll hear from Rachel about the EBI itself, the mechanisms through which they support interdisciplinary research, including via the funding of research strands such as this one. Then Emma Vincent's going to give you a little bit of an overview of what this strand's about and how you can engage with it, and how to shape the direction of the strand so that it can provide a useful hub and resource for people wanting to engage. Ruth Mitchell will then present further information on how to engage with the strand and give you some examples of the sorts of projects that can benefit from these ways of working. In the next week or so, we're hoping to send out a survey to those of you who've agreed to be on the mailing list to ask us what you hope to get out of the strand. Please do take a couple of minutes to feedback to us, because the more we know about what you want, the more that we can tailor our help, training and events to fit that and promote interdisciplinary research. So with further ado, I'd really like to welcome Professor Guberman-Hill.
Fantastic. Thanks so much for that really nice introduction. So I'm Rachel, and I'm director of the Elizabeth Blackwell Institute. And what we thought might be really useful before we kick off today was just if I say a few words about the Elizabeth Blackwell Institute and how we work and how we support research across the university. I won't keep you from the main business, so I'll keep this fairly short. So in brief, the Elizabeth Blackwell Institute is one of 5 university research institutes. And as a University Research Institute, what we do is we support and nurture research across all faculties, and with a particular emphasis on bringing disciplines together to work together to build on the excellent research that we already do and to help researchers move their projects to the next stages. At the Institute, we're really proud to bear the name of Dr. Elizabeth Blackwell, and most of you probably already know that she was the first woman on the British Medical Register, and she was born just down the road in Bristol. So the way in which we work at the Institute to support research across the university in health is by running funding schemes and by setting up and supporting research initiatives. One of the key ways that we've been doing that in recent years is through thematic research strands. And I'm really pleased to say that Mechanisms to Populations is the 7th research strand that's been set up. And the aim of the strands is really to connect the excellent, really high quality research that's already going on. So today I'm really excited to learn from you all about what you think the research strand can be doing, and to hear from you about the next steps for the strand, and also to learn about the research that's happening across a variety of disciplines. In normal times, we'd provide you with a relatively decent lunch and certainly a decent coffee. So I'm really sorry that we can't do that for you today. But I thank you all for coming. I know everyone's massively busy, so it's really fantastic that you've made time for the conversation today. And thank you in particular to all the organisers for making this happen. So well done, and this is going to be a great event. Thank you very much. I'm going to hand over now to Emma Vincent, who's going to introduce the strand in more detail. Thanks, folks.
Okay, thanks, Rachel. I'll just share my screen. Okay. Yeah, it's a real pleasure to welcome everyone today to the launch of this Strand. And Chrissy and I are particularly excited about it, to see it come to fruition. And we do really hope that it is going to be genuinely useful to people. So we've covered housekeeping. So I'm going to skip straight on into introductions. So this is the team for the Strand, integrating health research mechanisms to populations. So Chrissy and I co-lead this Strand. We'll just introduce ourselves very briefly. So I'm a Diabetes UK Research Fellow, and my fellowship and research group are working across both the schools of cellular and molecular medicine and population health science. And my appointment's actually split between the two schools. So my background is in cell biology, but I started working with the MRC Integrative Epidemiology Unit in 2016, and which at the time was like learning a completely new language. But it's been an incredibly valuable journey for me and for my team since then. And we now publish papers in both disciplines, and we've recently started to publish sort of truly interdisciplinary papers with a contribution from both disciplines, which has been proving really impactful. And so I guess it's because of my experience that I'm really invested in making interdisciplinary working more accessible for researchers at the University of Bristol. And that's obviously led me to where I am now with this Strand. Right, so I'll just hand over to Chrissy briefly to introduce herself to you.
So like Emma, I also benefit from close interactions with people in population health. I'm a senior research fellow funded by Versus Arthritis over in the School of Physiology, Pharmacology and Neuroscience. And while I'm more embedded in the biomedical health side of things, members of my group have really benefited from working closely with people in the musculoskeletal research unit up at Southmead Hospital and in other population studies beyond that. And one of the things that, like Emma, we're really keen to do is to just help people to bridge the gap between the two disciplines, because certainly for myself, I knew that when we started out, I found finding the right people, speaking the right language, finding the best ways to work together, slightly challenging. And so we wanted to try to make that as simple as possible. And that's what we're hoping to do with this Strand. And apologies, that was my cat peering through the cat flap.
Thanks, Chrissy. So we've also got Ruth working on this Strand. So Ruth is our senior research associate on this research Strand, and Ruth has experience of working across both the disciplines of population health and the more biomedical sciences. And I'm not going to talk too much more about this here because Ruth's going to introduce herself to you and her background very soon. And also we have Esther working on Strand administration. And just to say thanks to Esther for doing such a wonderful job of organising and pulling this event together today. So thanks, Esther, for all your hard work, and also for the other members of the EBR team that are helping us out today. So that's the team. And we kicked off on the 1st of April, and we've got funding to run for a further 2 years. So what is this research Strand all about? Well, it's about facilitating research and collaboration between the disciplines of population health science and what we're calling discovery science, but essentially fundamental bioscience or laboratory-based research. So interdisciplinary research is valuable and it's impactful, and I'm sure I don't need to particularly sell that to you today. But population health science can provide increased human and clinical relevance to laboratory findings, so moving beyond sort of in vitro and animal models. And in turn, discovery science can provide improved functional relevance and validation to population health science findings. So, for instance, providing functional relevance to GWAS hits. So they're two disciplines with the potential to really complement each other and enhance the work of each other. And we also really wanted to make the point with this figure that it can work in both directions. And we really want this research Strand to benefit people on both sides and in both disciplines. And Ruth is going to give some nice examples of that in her talk. So being able to recognise the power and advantage of interdisciplinary research is relatively easy, but actually undertaking it is often not. And Chrissy and I have always felt that there is great enthusiasm for interdisciplinary research in this space, but there are barriers in place to doing it that often mean that exciting projects don't get off the ground. So some common issues and some common barriers are a language barrier, and we hear this comment a lot from people working across the disciplines, that it is like learning a new language. And so this barrier, it can be particularly where a lot of projects sort of fall down or don't get off the ground. There's a sort of feasibility barrier, or I guess an understanding of feasibility, which can mean that projects sort of don't get started. Skills barrier is a big thing. So having access to people with the skills to take on the work that needs doing. In the same way, time, having people with time to dedicate. But also there's a different understanding often of time scales across the disciplines. And for instance, a genetic epidemiology paper might take months to put together, whereas a cell biology paper might take years. So this is often an issue as well with managing these sorts of collaborations. And expectations as well. So understanding of expectations, understanding what can be achieved. So then what do we think is needed? And in turn, what can this research strand offer in order to break down some of these barriers? So firstly, and really importantly, we now have a home for interdisciplinary research in this space. So a contact point and a means of communication. So we have a website and email address that we've got set up now. And so there's now a point of contact for people who are interested in working in this space and so can start to explore possibilities. We decided that what was needed above all else was access to researcher time and expertise. And this is why we've decided to devote the budget of this strand entirely over to salary. We decided that having a researcher in place with the available time, experience in both disciplines and skills working across both disciplines to be able to translate and work with and facilitate collaboration. We decided that that was really going to be crucial to making this work. So we've obviously got Ruth in place to facilitate this, and Ruth will be able to discuss your needs with you, the type of projects that you want to do. And this might involve her undertaking perhaps a discrete piece of work, say some preliminary data for a grant, or help write a section of a grant or a studentship or a fellowship. But it might also take the form of sort of matching or forming collaborations for those longer term projects that are sort of more slow burner projects. So Ruth will be the first port of call for discussions, but we also have an excellent steering group attached to this strand and many more interested parties that we can draw on. But this is also where we're keen to have as many people involved as possible. And we're going to send a survey or questionnaire out after this event. And so if you are interested in getting involved or happy to be contacted about collaboration or working in this space, then please do say so on that questionnaire. The third really important thing that we thought was needed was community building in the space between the two disciplines or the two sides. So, you know, there are several sort of discrete collaborations that are going on in this space, but I would say that there's little in the way of sort of a joint community between the two. And so Ruth is going to work on particular events and workshops and training that will help build this community. And again, this is where we really would like your input on what you want the strand to do for you. Sort of what events would you want? What training or workshops would you want? And so please do get in touch and use that questionnaire that we'll send out after this event to let us know what you want. So we considered that investing in these three things was the best way to break down those barriers that I talked about on the previous slide. And we thought this was probably also the most inclusive way to manage this research strand, to hopefully benefit as many people as possible, and particularly early career researchers, which we feel can really benefit from this strand and that we hope will. there's obviously going to be some kind of capacity. Ruth at the end of the day is only one person, but we will work together as a team. We do have the steering group and others to maximise the most out of this. And importantly, if this is successful, if it is what people want and what people need, then we can think and plan for longevity beyond this 2-year funding period. So I've put this slide in just to sort of demonstrate some examples of how you might engage with the research strand. Obviously, this is completely flexible and it's just a demonstration, and it's not necessarily how people might engage or the order, but just to give some sort of examples of how to engage. So it might take the form of sort of community building events sort of at the start of the project to get a feel of who's out there and what's possible. You might engage with the strand to initiate a collaboration to help identify sort of like-minded parties. And I sort of want to stress here that, you know, even if you don't have a particular project in mind, but you just want to sit down with Ruth, discuss possibilities, then absolutely do that. Don't feel you have to have sort of a set defined project in mind in order to engage with the strand. So we might also be able to help submit an interdisciplinary project proposal. This might be for a doctoral training programme, for instance, or a PhD rotation mini project. And as I mentioned earlier, it might take the form of perhaps a discrete piece of preliminary data that you might need for a grant. We also would be able to provide sort of letters of support for grants as well. So to just demonstrate that resource is in place. It might also involve a bit of a discrete piece of data to enhance the impact of a publication, for instance. Or, you know, it might take the form of training, and obviously training could take place at any place in a project, but I've sort of put it towards the end as a sort of demonstration that you might want to have some particular longevity in interdisciplinary working and therefore want yourself or your student or postdoc to be trained in those skills. So as I said, please do engage with us. That's our email address, and we have the website. And if you signed up, you're probably now also signed up to the mailing list. So please do fill in that follow-on survey so we can understand a bit more about what people want. But to finish, just to say thank you for coming along and to finding out more about the Strand today. And obviously thank you to the EBI as well for funding this Strand. So I'll hand back now to Chrissy.
Thank you very much, Emma. And what we'll do is move pretty swiftly on to Ruth, who's going to give you some examples of projects that have worked and the sort of tools that one might use in starting those conversations, thinking about how this could fit into your own research or research you might plan in the future. So I'd like to introduce Dr. Ruth Mitchell.
Thank you. I've managed to share my screen. I didn't manage to put my video in. There we go. Great. So as Emma and Chrissy have said, that we're very grateful to the EBI for funding this, and as they are both very excited, I'm very excited to be working alongside them on this research strand of integrating health research and mechanisms to populations. And Emma has already given some of my background into this, and I just want to go a bit more into that, a bit more into some ideas for that. So I'm going to give a bit of background about me, give some ideas of how we envisage integrating what we're calling fundamental bioscience, but as Emma has said, basically laboratory science and population health. My two experiences of this, and you will see this, are cell biology and epidemiology. And then give some ideas of how you guys can engage in the Strand, and basically to say, as Emma has already said, we're here for you. We're here to facilitate interdisciplinary research, and therefore we very much value your input into this and what you want, and we'd love to be able to facilitate collaborations, break down some of the barriers that Emma has mentioned. And I'm going to finish off with some examples of some outputs that we envisage for the Strand. So just to start off with, I did my PhD in cellular and molecular medicine with David Wraith, and this was in immunology. I'm an immunologist and interested in the immune system, and this will be evident throughout the different projects that I have carried out. And I'm going to show bits of science, but it's not so much about the science. It's mainly just to show you that I've worked in cell biology, and we were working on a mouse model. So we did in vivo and in vitro work. And essentially what we did was, using this mouse model of multiple sclerosis, we looked at Th1 cells, so cells of the adaptive immune system. And if you give them successive treatments of antigen, then they become anergic and no longer cause disease and start secreting interleukin-10, which is an anti-inflammatory messenger. So in a couple of examples of some of the work I did, I used in vivo mouse model of an IL-4 receptor knockout. So I was very interested in how IL-4, so interleukin-4, a messenger, regulates interleukin-10, this anti-inflammatory cytokine. And we can see that without IL-4, you do not get this upregulation of IL-10 as seen with it. And I followed this up with many in vitro experiments, but one of them was looking at a co-culture, so using Th2 cells as a source of IL-4, as they naturally secrete IL-4, and looking at the effect on Th1 cells. And we can see that the Th1 cells upregulated their IL-4 receptor, and they phosphorylated STAT6, so started IL-4 signaling. And again, when you injected these back into the mice, they no longer caused disease. So that's essentially giving a bit of background into my cell biology background. I then transitioned post-PhD into epidemiology, into the integrative epidemiology here in population health sciences in Bristol. And this was on a postgraduate discipline hopping fellowship funded by the EBI. And during that, I was working with Lavinia Paternoster, who very kindly took me on and taught me what I know about epidemiology and genetics. And then George, I'm then a postdoc with George, and I'm continuing with that. And Georgia was very gracious. For the last 6 months, I've been working with Nick Timpson on national core studies and their response to the COVID pandemic. So in epidemiology, we essentially look at an exposure and an outcome. And we want to know the association, we want to know causality in that. And again, sat here thinking about it, you can basically replace exposure and outcome by your protein of interest and your disease of interest in that. In epidemiology, we use multiple study designs to do this. So a cross-sectional study, so looking at a point in time, and this can tell you a prevalence of a disease, a case control study where you single out cases of a particular disease, cohort studies, which are usually longitudinal. And so you collect a wealth of data, and they're just amazing to mine for the exposureness and outcomes that you can look at. And as a gold standard, we have a randomized control trial, which, because of the randomization between the groups, and then you have an intervention and a control group within that, this takes away the confounders that can affect your exposure outcome association. So just to give you some examples of the studies that I have experience of working with. So ALSPAC, the Avon Longitudinal Study of Parents and Children, also known as the Children of the 90s, is a Bristol-based cohort, and we essentially recruited children in utero and their mothers and have followed them up for nearly 30 years now. And so this has then become a multi-generational data set and a longitudinal data set. And this resource is there, it's Bristol-based, it's something that the ALSPAC team are very much open to suggestions of going back and recruiting more people or looking at particular traits that they already have. This data is there, it is there to be mined. Another source of data is UK Biobank, and this is an adult cohort of 500,000 individuals. And this is, they have genetic data. So it's an amazing resource of genetic data, and then with biological measurements, lifestyle indicators, biomarkers in blood and urine, and imaging of the body and brain. And so you can see that both of these resources are then there to be used, to be mined. They are open, open for that. So just to give you some of the ideas of the projects that I've been involved in, or some of the ideas that are possible to do with this sort of data. The MRC-IEU is focused on genetics, and so the projects I have done are very much focused on genetics, but this does not undermine the fact of doing a non-genetic study in population health. So I started out with Lavinia looking at multiple sclerosis PheWAS, so a phenome-wide association study. And I did this in UK Biobank. So we took from the International Multiple Sclerosis Genetic Consortium the genetic variants that were associated with MS. And essentially in each individual in UK Biobank, I generated a genetic risk score for MS, and then we correlated that to the phenome. And I did this through a tool called Pheasant, which Louise Millard developed in the IEU. And it's just to say that this then, for example, eosinophil count and percentage were pulled up as interesting, and these will then be followed up into further studies. So another thing that the IEU is mainly known for with George Davey-Smith is Mendelian randomization, and I highly recommend going to watch his 2-minute primer on it. I'm not going to show it here, but essentially, again, we're looking at exposure and outcome, but the problem with this association is obviously the confounders in it. And so MR, Mendelian randomization, which I will call MR, takes the genetic variants that are associated with the exposure and proxies the exposure using them, and then looks at their association with the outcome through this, all working through the exposure. So one of the beauties of this is you can obviously do this in cohort studies such as UK Biobank and ALSPAC, where genetic data is available. But two-sample Mendelian randomization has opened this up as any disease or any exposure or outcome, so trait and disease of interest, as long as there's a publicly available GWAS, you can perform Mendelian randomization in that. And this is essentially showing that you would take the genetic variants associated with your exposure and look them up in this summary, what we call the summary statistics of your outcome GWAS, and to look for the causal association between the two. This has been automated in MRBase, and this is also a massive resource in terms of a pool of all the available genome-wide association studies that are available. And this is there. It's a web-based portal that you can go and you can explore. So as an example, I've been involved in an MR of neutrophil count and malaria, and this is through collaboration with Borko Alumnek in cellular and molecular medicine. And essentially, as any collaboration starts, I presented at a meeting and he came and approached me, and his lab is focused on malaria, and we decided to try and implement an MR of this using the genetic data available. So we then need to go and find genetic instruments for neutrophil counts. And as I said, usually this is done through looking up a genome-wide association study that is publicly available. However, these tend to be in European populations. And as we know, malaria is not a European-based disease. So we decided to use the African population in UK Biobank, and David Hughes was instrumental in helping us identify these through their genetics. And we then performed a genome-wide association study to find those instruments. For the outcome of the disease of malaria, there has been a malaria susceptibility GWAS performed. And so we used their summaries, the publicly available summary statistics. So when looking in the direction of neutrophil count on malaria, we don't see any association. But when the beauty of MR is you can perform it in both directions. So this is now a genetic liability to malaria, and we can see that it has an effect on severe malaria in particular. So something I've mentioned is that this was on, for example, susceptibility of malaria, and a lot of the GWAS, genome-wide association studies that have been performed are on case control studies, and therefore are to do with the incidence or to do with susceptibility. However, we are moving into a phase of performing these in progression. So in cases of those disease, this throws up a whole new area of bias, but this is an active area of research, and this field, the field of genetic epidemiology, rapidly progresses, and so therefore this is coming. And so you can see with incidence, we were looking at causes and potential mechanisms for prevention, but however with progression, we're looking more for effective treatment of that. So I now just want to give you some ideas in genetic epidemiology of how we can integrate bioscience, fundamental bioscience. And so you have your GWAS hits, which are genetic variants that are associated with your disease of interest. You can look them up and see what gene they're involved in, and then what tissue they might be expressed in. And then the ultimate aim is to be able to prevent this, to be able to find a drug to treat this. And so essentially we have population health sciences and discovery science, and both working towards the aim of improved patient health. And as Emma said, this is definitely a two-way conversation between the two. It's not about one informing the other, one holding the high ground of the other. We need each other to be able to do this. So in population health sciences, as I've said, we look at exposure and outcome, and we question the causality between the association. And then a causal trait, causal gene are then taken into further analysis. So the advantage of this is obviously we're now looking in a whole body system. We're looking in human populations, and this can inform laboratory science. On the other side, we have fundamental bioscience, lab-based laboratory science. And you guys look at pathways, proteins, model organisms at disease or trait of interest. And this is actually extremely difficult to instrument and to get down to the nitty-gritty of each molecule in a whole body system. There are obviously ways we can do that. There are ways that we're limited with that as well. And therefore, yes, we can look at a whole human system as we do that, but we also need you to inform our studies as the other way goes the other way around as well. So I've mentioned particularly disease GWAS, but there are also protein GWASs. So this is the plasma proteome of human metabolism, and interestingly for me, the cytokine GWASs. And so these are up and coming, which means that your exposure can then be replaced by a protein, as it were. And an example of this, and one which I have experience of working in, is a cohort in Paris called the Milieu Intérieur project. And they've collected about 1,000 healthy individuals and then done deep phenotyping on their immune cells down to different surface cell markers so that you can differentiate the different immune cells. And therefore, because of their other deep phenotyping with that, it's possible then to look at those cells in relation to a trait, a demographic trait within the population. So something we can do, and these web portals are available, is, for example, thinking about a gene in bioscience that you are interested in, is then to look up the SNPs within those genes, and you can do that in published GWAS. And then particularly highlighting GTEx portal, which gives you the tissues of interest that that these genes are expressed in humans. So as an example, I've pulled out IL-10, the anti-inflammatory messenger cytokine that I mentioned earlier, and promisingly we see that it's in lymphocytes, and this can also give us single tissue expression eQTLs. So again, you can see that it's in whole blood, but then these are the genetic variants that are associated with the expression of that gene. So building on that, there's also being performed transcriptome-wide association studies, and this integrates GWAS and gene expression data sets to identify gene trait associations. So now no longer just looking at your genotype and your phenotype, but now looking at the expression in your phenotype. And another way of integrating expression, quantitative trait loci, and GWAS data is through co-localization, and this can bring up target genes. And just to say on this, we're hoping to populate the website, the EBI Strand website, with resources such as these to be of help that are available to look at. And if there are others out there that you have found useful in your own work, then we'd very much love to hear about that and populate the website with them. So another way of doing Mendelian randomization is tissue-specific MR. And Tom Richardson has done this by applying Mendelian randomization and genetic co-localization in this. And again, his web app is available to look at. Another great wealth of resource is the Epigraph DB. And this was the proteome PheWAS browser. So looking at the phenome, and I pulled up interleukin-23 receptor, which is associated with inflammatory bowel disease. So it's just showing you that you can then associate a protein and a disease within data sets that are available. So then going back to that, I've shown you a bit about how we can integrate population health sciences and discovery science. And essentially this is very important. And as Marcus Manafo and George David Smith have talked about, we definitely need triangulation within this. We need different lines of evidence in improving patient health. We need each other, and we need to start talking to each other. And as Emma said previously, we're hoping that this strand might be a point of contact, a point of call in facilitating those. So then some of the ways of engaging with the strand that we've envisaged, I've put essentially three down here, but this is, we mainly want to work with you, and this will also depend on what you want. So please, we'd love your feedback, and in signing up for this, we realize that you've already given us feedback, and we're very grateful for that. But essentially, we want to help you and help you facilitate interdisciplinary research between population health sciences and fundamental bioscience. So some of the ways of engaging in the strand would be a discrete piece of work. And I think initially for this 2-year period, we're possibly just going pieces of work in terms of population health sciences work, rather than me myself going into the labs, because you guys know much better about that. So that could be a piece of work to go into a paper, a piece of work to go into a grant. But we're also here to facilitate collaboration. But in saying that, we are aware of collaborations that are already ongoing. And essentially, we want to encourage that. We don't want to take away from that at all. We want to facilitate that. And then there'll be community building events and potentially one-off seminars if that would be of interest to people. So essentially, in engaging in population health sciences, you need an exposure and an outcome, a gene you would like to follow up going the other way into bioscience. And we can sit down and have a conversation about a potential collaboration. So some example outputs are contributions towards papers. And this is a beautiful example from Emma, where Emma and Caroline went to a conference and they presented Mendelian Randomization study looking at LDL cholesterol and renal cell carcinoma, where we can see that increased circulating LDL levels are associated with increased risk of renal cell carcinoma. And we can see that this is LDL receptor expression is increased in the tumours as well. And there was a poster there that was actually looking at cholesterol in mouse models. And they've pulled their data, and this has now been accepted for publication in Cancer Discovery. So another example would be a team collaboration, and this is part of the work that I mentioned earlier, looking at the cohort studies response to the COVID pandemic. And this is through the UKCIC, which looks at the immunological response to COVID. And this was using data collected through ALSPAC. And so through questionnaires, we defined self-report of COVID. There's serology, which was sent out with the 3rd COVID questionnaire. And then we have linkage to the Public Health England testing that is happening. And with a combination of this data, we defined cases, and we've called these cases back in together with matched controls and random controls into repeat clinics, where measurable traits will be taken, but biosamples will also be taken, and these are being sent to the labs in cellular and molecular medicine to look at their T-cell response and their antibody response, and there will be some pseudovirus work as well going on. but essentially we can use the wealth of data that ALSPAC has from these people, not just at these snapshots in time in these clinics, to then look at their response to a COVID infection and the immune response mounted through T cells and antibodies. So then another example would be of a grant application, and this is with Dylan Bergan. This is his fellowship application, and essentially he's using genome-wide association studies, and he's doing an in silico prioritization of genes rates to then go on and look at functional studies in zebrafish, but essentially he's using the GWAS studies to inform his fundamental bioscience studies. So then I hope I've shown you that these work both ways, both in terms of population health sciences informing lab studies, but as well as lab studies being used in combination with population health sciences. And we just have a little poll now. We're going to go into a Q&A session about this, but just to get our thinking caps on and get us thinking about this, we have 3 questions. So what department you're in, what stage of collaborating you're at, and what sort of engagement with the Strand you guys would like. So I'm just going to put the polls up now. And if you could answer those, that would be very helpful. Thank you. So as Emma already highlighted, there is the email address for any questions. I'd love to sit down with you and have a conversation about collaboration. And even if that it isn't just a discrete piece of work that I will be able to help with my time on this Strand, it could be simply introducing you to the right people. And we want to build up a database of people like yourselves who are here, who are open for collaboration. And again, there'll be the website, and we're hoping to populate that with resources as well. Great, thank you. I'm going to end the poll there. That was very helpful, thank you. And this brings me to the acknowledgements. And of course, this is the EBI, Emma and Chrissy, working alongside them on this Strand. This is essentially my journey through research, and so my PhD supervisors, and also then my postdoc supervisors and the integrative epidemiology unit for all the help that they've been as well. And I believe I'm now handing back to Emma and Chrissy, and we're now in a Q&A session.
Thank you so much, Ruth. That was excellent. I hope that's given people a bit of food for thought about the sort of ways in which you can engage. And mostly we wanted to use this as a prompt to just start to think about how we can help you. So what we want to then know from you is, what do you need? What do you want? How can we best get this to be as functional and helpful to as many people as it can be? As I said before, we're particularly keen to engage with early career researchers, the people who might not know so many people around the 2 departments. We really want to help bridge that gap between the different areas in the university. Please do stick in questions, otherwise you'll end up with us just waffling for some time. So we've got one from Caitlin. To add to the excellent summary of what Mendelian randomization is, anyone interested in learning more about the concept, please see our new online dictionary, MR Dictionary at mrcieu.ac.uk, and the MR short course. Thank you very much for posting that. That's incredibly useful. That's another thing that we would love from you, as well as telling us what you want. What do you want on the website? Tell us of other resources you're already aware of, other links to good YouTubes, easily accessed and informative, like primers, websites that have like worked examples. One of the things that Ruth's hoping to do is to also build up some sort of like how-to videos over the duration of this strand. So again, knowing what you'd like to get going with would be really helpful. So if you want essentially like an example of how to do and use MR base for it as an example, but please, yeah, do talk to us. Got a question from Luca. You talked mostly about 2 areas of 2 departments. We're looking for the help of mathematical modelers. Absolutely. We're really interested in engaging anybody across. What we want is to just be able to get the best out of what's already available to a degree. So yes, absolutely. If you are a mathematical modeler and are looking for ways to engage either with fundamental biosciences or population health, yes, please do. Please come and talk to us, essentially. Tell us what your own specific questions or interests are. Then one of the things that we hope to do is, by building this community, almost do a bit of like help people speed date a little bit between disciplines, help them find people who can help them with the specific questions that they might have. There's a question from Hannah higher up. Maybe a silly question, but does the scheme cover links with bioinformaticians? Absolutely not a silly question. Yes, we would the idea is to just try to help to link up people wherever absolutely possible.
I could tell you all, since I'm now here, I can't seem to turn my video on either, actually, but I could tell you one way in which this program's already delivering. I mentioned to several of the committee yesterday or a few days ago, I begged an early go on this because Helen Weavers and I had a grant application we were putting in, and we really needed 2 or 3 lines at the end about how our fly studies going to mouse studies could lead on to some population health insights. Ruth knocked us together 3 or 4 lines, and we got the program grant a few weeks ago. So that's sort of 1 program grant in the bag for the Strand already.
Really good question from Lucy McGowan. Are we planning to hold grant writing workshops or networking events for people who need to bounce ideas around? Yes, absolutely. One of the things that we'll ask with follow-up stuff is essentially what sort of time frame are people working to? Because one of the things we need to work out is when to hold stuff. So to have a bit of an understanding of how imminently people need things to happen, what stage of ideas people are at, and then we can work out essentially how to hold more targeted events to help people get going. So yes, absolutely, that is exactly the idea to this, is to sort of help to host some sandpit events, help people get together. But what we do need from people is an idea of what they want and when, so that we know, are we essentially rushing to hold it as soon as possible, or are we building up a larger network to hold it at a time that's most useful? A really useful comment, which is considering how people here can disseminate information about the existence of strands and opportunities. Yes, please do pass on information. Please particularly encourage early career researchers to engage with us. We will be in contact to ask people, again, in this sort of follow-up survey, like what degree of willingness have you got to help follow this, help out, whether people are keen to become ambassadors, whether people are keen to engage as potential collaborators, to give further information about other things, like whether that's a model organism that they work on, whether that's a particular resource they work most frequently with, whether that's something like MR base, or in my case, zebrafish, or in someone else's case, cell lines. Yes, absolutely. The thing that we're most keen to is to spread this to be as useful to as many people as possible. Lindsay's put up a brilliant link to an MR explainer, which is in the chat. So for people looking to get going with Mendelian randomization, in the chat, there's a link which we'll also add to the website, which is written in lay terms, which is always particularly useful. One question that we will follow up with after this is whether people would prefer future events to be online or to be held in person or to be blended, because we're now at the sort of stage where we've been forced into online, obviously, to get going, but that model can begin to change. And so understanding a little bit about what people's preferences are would be really helpful to us. So we will follow up with that as well to get a better understanding of what would work for people. Is it small networking events, or is it larger scale things? Larger scale is more likely to be online, or would people value essentially the sort of the chat over a cup of coffee that like lets people really run through their ideas in a sort of a sort of a more social setting, I guess. Emma, anything you want to add to that?
No, I don't think so. I think, yeah, you're right. Like it's just going to be really important for people to tell us what they want to do, what they want to see, and we'll do our best then to fulfill that. But yeah, I think just like I said in my talk, just even if you don't really know how you might engage, do just have a conversation, have a chat with Ruth and explain what you do, where you think you might be able to work in the interdisciplinary space, and because there's often a lot out there that, unless you sort of take the plunge and explore it a bit, so do just get in touch, get engaged. Yeah.
And we're aware lots of people will have to rush off, on the hour and things to, other meetings, to other stuff. If there are questions you're not comfortable asking in a larger group setting, pop an email to us via the website, or to us personally. You've got our details via the website too. What we really want to do is to just talk, get the conversation going. That was essentially the idea of today, is to just use this as a beginning to this. Just to give people some ideas and start people thinking about it, so that over time people can engage at whatever time feels right. So a question about, will a list of experts be generated, posted to facilitate bilateral collaborations? Absolutely. One of the things we'll follow up with when we ask is are you willing to be contacted? Because we don't want to essentially throw people in at the deep end by saying these people are working this if they're already swamped and don't have the capacity to do so. But yes, absolutely, we would love to essentially populate a resource that says who works on the people who are willing, and what do they work on, how might they be prepared to engage. And that's something that we can then build up an area for in the website too. But yeah, for anybody who has to disappear off, thank you so much for coming along. We appreciate it's an incredibly busy time with exams about to kick off and everything else as well. So yeah, thank you very much for coming along. A relatively short notice when we got this started. I hope it has been useful to people. And we really hope that over the course of the next 2 years, we can really build on this and turn it into something that helps the most possible people. And I'd like to sort of also finish off by saying thank you again to the EBI. It's only through their generous support that we're able to actually put a person in place who can really help turn things from vague ideas that you might have had and not known where to go with, and really help that to move forward.
Chrissy, if I may, that's super kind, but it's only down to all of your work that it's happening. So the thanks really are due to you as a Strand team.
We'll bask in that glory. It feels like we've hopefully answered the questions that have come up, but please do keep asking us, and at any point